Okay, everybody, so I'm going to just cover the last two sections here in this one video presentation. This is 7.4, using a spectrophotometer to measure protein concentration. So this is just the deal that we've been doing forever in biotech, okay? So basically how a spectrophotometer is just another way to measure protein concentration. Let's look at the importance of measuring the absorbance spectrum and lambda max of a particular protein. Explain why the spec would be set at 395 nanometers in order to detect hemoglobin in solution. Explain why a UV spec would be set at 280 nanometers. Explain how protein indicator solutions are used with and without a spec. Describe the use of a Bradford reagent and others such as Fadibus tablets. Define and describe a standard curve. Be able to use the best fit standard curve to determine the concentration of an unknown protein sample. Explain how an ELISA plate reader with a built-in spec works. Okay, so a spectrophotometer is just another way to measure protein concentration, and then the importance of measuring the absorbance spectrum and lambda max of a particular protein. Now, I'm going to be doing that with the first thing that I'm going to give you for the little lab, the Chapter 7 labs that I'm putting together. I haven't done that yet, but there's going to be a little thing that'll be set up tomorrow morning that I've comboed. And I found a cute thing that I hope will be okay. I think it's still a flash player thing. I hope everybody can figure out how to do that. But it's cute because you can do the whole lambda max and concentration of red and blue dye and dye solutions. Anywho, you'll see me also do the Bradford one. So let's just walk it through here. So 7.4, so protein concentration activity measured by ELISA page activity assays, but a spec determines protein molecule concentration because of its direct relationship to light absorbance, back to Beer's law. Absorbance equals molar absorptivity times length of light path, times concentration in moles per liter. So you are getting your absorbance spectrum from a protein by determining measuring its protein light absorbance at different wavelengths. You scan it from 400 to 700 nanometers if it's colored. Otherwise, if it's UV, you would do it from 200 to 350. Lambda max is a wavelength at which the greatest amount of light absorbance occurs for that particular protein. It's the peak of the molecule's absorbance spectrum. And again, we go back to the color wheel. If it was a red solution, then you're going to be looking at the green. And then if it's blue, you're going to look at the yellow. So it's going to be that kind of complementary color thing where you can see like what you can predict where your lambda max is going to be. Okay, why would the spec be set at 395 nanometers in order to detect hemoglobin in solution? Okay, well, hemoglobin is red in the presence of oxygen, so it absorbs blue light waves, complementary color, blue-green area there. Hemoglobin's lambda max is 395, so the spec would be set to that wavelength, so any hemoglobin molecules that were present in solution would be detected. Why does the UV spec set at 280 nanometers? Okay, if you set this wavelength at 280 nanometers in a UV spec, again, we're working with things that are colorless. They have no indicator to them. They're see-through because many amino acids absorb light at 280 nanometers. So the lambda max of protein without an indicator, completely colorless, is 280 nanometers. It's sort of like a memorization thing. You have to memorize that. So it is easy way to determine the total amount of protein solution, but not a good way to detect certain protein solution. That's when we have to do Page and Western blot to do that. Okay, how protein indicator solutions are used with and without a spec. Describe the use of a Bradford reagent and others such as Fadibus tablets. The only way, and this is Bradford, so Bradford's sort of the standard, it's the go-to protein indicator. So it's a way to make colorless proteins have color. So the only way colorless proteins can be detected in a vis spectrophotometer is use an indicator. So that's Bradford. So Bradford reagent changes from brown to blue in the presence of protein. The darker blue samples mean a higher concentration of protein molecules and therefore absorb more light. Bradford testing is often used to calculate protein concentration in unknown samples. Unfortunately, I don't think I'm going to be able to do that. I have protein. I have to see what's going on. I have the standards. I'll see how it goes. Oh, I have a protein assay kit. Yeah, I will be doing it. I think it's Bradford. I have to look back and see. All right, define and describe the standard curve. Be able to use a best fit standard curve to determine the concentration of unknown protein sample. Okay, we've been doing this since Biotech 1. So with copper sulfate, you would do it, and then you would just do because you're working with making different kinds of solutions. But this one is that we've done it, some kind of zero dilution of a stock solution of something, and then it's a way for you to get a standard curve, and then you can go ahead and solve by using the equation of the line and get the absorbance. So let's walk it through. So basically, make a standard diluted protein samples of known concentrations and mix them with Bradford reagents. So, whether you do a stock and then you just do a 1 to 2 dilution or 1 to 5 or 1 to 10, you're breaking it down into known concentrations. Then find the absorbances of these samples using the lambda max. That's the wavelength of the undiluted protein. Like where was it? What wavelength did it have maximum absorbance? That's what you set your spec on. And then the Bradford reagent. Then once you get those absorbance values, plot them on a graph comparing concentration to absorbance. Make a best fit or trend line. That's called the standard curve. So that's always been a standard curve. The trend line, the R squared, the trend line, the R squared. Now we've been doing other things. We've been doing log trend lines and all that, logarithmic trend lines or exponential trend lines. We're not doing that on this one. This is just very basic. This is linear, so it's very simple. Just a linear trend line with an equation of the line. Okay, y equals mx plus b. Okay, so then comparing the absorbance of unknown mixtures with the absorbance of the known concentrations on the standard curve, you can estimate the number of protein molecules in a given volume solution. So then you have the equation. It's going to be y equals mx plus b as you see it, on the slideshow program, how you do that, whether it's Google Google spreadsheet or an Excel. Then you have to flip it and manipulate it for x because y is the absorbance. So y is the absorbance. So you would plug it. So it's x, which is concentration, equals y, the absorbance, minus b, the slope intercept, over m, the slope. So y again is the absorbance from the spec, and x is the concentration of your sample. So that's like, I'm snapping my fingers, snapping, because that's something we've been doing a lot, and you've been doing it quite a bit. Okay, so ELISA plate reader is like, oh, well, we couldn't get to that this year. So if anybody wants to do science fair in the fall, we might be able to do it. I was trying to get to it at some point, especially with the Western blot and the continuation of the ELISA, but we do have one. And it's basically using a well plate, plugging it in, getting the absorbance readings of everything that's in it. So you can do that. So a plate reader basically is a way to take a well plate, like a 96 well plate, and then just put it in, or a 24 well plate that we have, what have you, and put it into the box, and it'll, give you a readout of all the absorbances in what you have. So it's really, it's very efficient that way. Now, if you have to detect a particular protein solution of many, of course, ELISA using antibodies to detect certain proteins. You can use Phaedovis tablets. They change color in the presence of amylase. An ELISA plate reader is a built-in spec. It quickly reads the concentration and colorimetric ELISA samples in each well of an immunological ELISA well plate. So when we had done it, we just looked at the colors of the strips that we had in ELISA. If we had gone one step further, which I was going to, with all our strips, I would have plugged them in to the ELISA plate reader, and we would have gotten the absorbance readings. We could have done some standard curves. Okay, and then the review questions are in there. Okay, let's go right over to 7.5. That's using a spec to measure DNA concentration and purity. So quantification of DNA in solution is very important for many biotechnological applications. Genomic DNA and plasma DNA are routinely isolated for research and diagnostic purposes. So DNA is colorless, so the UV spectrophotometer is used. The lambda max for DNA in solution is 260 nanometers. So let's go back. The protein's lambda max is 280. The DNA's lambda max is 260. So those are just things you memorize. Protein's 280, DNA's 260, and this is all in the UV range. So that's what's going on there. So DNA concentration can be determined by measuring the optical density, OD, or absorbance of the sample at 260 nanometers. Maybe at some point you had heard about that when we talked about bacterial growth last year in Biotech 2. You would hear of OD as optical density of your broth culture that you would do, and that could be equated out. You could get the value of that, absorbance values, and do the same thing with finding out how much bacteria has been growing in your sample. So maybe you've heard that before on that note. Okay, now, optical density of DNA and concentration is an equation. If the optical density of DNA is 1 AU, 1 absorbance, then the concentration would be 50 micrograms per milliliter. So that's a conversion factor, conversion unit. That's it, like 12 feet in a, oh, I shouldn't say feet. No, not 12 inches in a foot. I shouldn't say that. 100 meters in 100 centimeters in 1 meter. That's it. That's all improvising here. So 100 centimeters in 1 meter is like 1 AU and 50 micrograms per milliliter. So it's a conversion unit. So by measuring the absorbance of a DNA sample at 260 nanometers, so you would just use a UV spec to get that, And knowing the relationship above, you can determine the concentration of DNA in a sample using the DNA concentration equation. Here it is. 50 micrograms per milliliter over 1 AU equals whatever it is, the unknown, micrograms per milliliter, over the sample's optical density at 260 nanometers, which is you're getting from the spectrophotometer. So you just get the absorbance values at 260, plug it into this equation, and solve for that. And that'll give you how much micrograms per milliliter of your DNA you have. Now, so if you did it with this, so you have the optical density at 0.32 AU, so you just plug it in, and then you go, oh, 16 micrograms per milliliter. That, again, we use the equation here to solve and to get that answer. Is this enough for a gel? Is it enough for a PCR? So DNA contamination determination is very important to pick out, too, because proteins are common contaminants of DNA samples. You're not fully extracting the DNA out. You're left with like some protein residues there because maybe you didn't add the protease or what have you. So proteins interfere with many reactions, including PCR and DNA sequencing. UV spec can be used to determine the relative purity of DNA in solution by assessing the relative amount of protein in a sample. So since the lambda max of protein is 280 nanometers, it can be compared to the lambda max of DNA at 260 nanometers in an equation to determine the relative amounts of DNA and protein in solution. So for optical density of 260 nanometers, optical density over that to 280 nanometers. So the numerator is your optical density of DNA, and the denominator is optical density of your protein. This same sample that you just put in that, you're just putting it in the spec, and you're flipping the wavelengths. So you get the absorbance at 260, write that down. Then you get the optical density at 280, write that down. And you put those two in the equation, and then you can put relative amount of DNA, a relative amount of protein, and then what's the degree of contamination of the sample. So you're going to look at the value of that equation. So the smaller the protein contamination is, the smaller the A280 reading and the larger the A260 over 280 value. If the answer comes out to being 1.8 or better, that DNA is relatively pure. So when you do your OD of 260 nanometers over the OD of 280 nanometers, and it comes out to 1.8 or better, your DNA is okay. If it's below 1.8, then the DNA has protein in it, and it needs further purification until it can be used for PCR sequencing. So if the answer is 2 or more, then the DNA sample has RNA in it, and that means you may need to run it through again with some RNases. If there's protein in it, run it through with some proteases, and that can clean up your DNA a little bit more there to get a better value. You want to get that 1.8. That's your pure, that's the number you want to get. Okay, these review questions you can do in your notes now. And then this is just T-Lab questions that were there at the end of 7. We don't have time really for me to add this on. So I just figured this would be good just to go through and see these and just check out the answers. I'll be composing some kind of questions, multiple choice questions in like this, related to this. So I would just go through it. Like this tells you how to work it, how are you making a buffer? So, that's brass tacks. We always want to know how to make a buffer. Okay, so make sure you're good there on that. And see another 2 sets of questions about that, how to make buffers. So for the content quiz, which I don't know when we're going to do that, we'll do it at some point near the end of your AP testing sessions. You want to be sure you can tell me how to do this. And these are just questions about what we were talking about with protein standard curves and doing concentration of proteins. And I'm going to have a couple things here for your labs so you can do that. Okay, that's it. All right, that finishes up the sections 7.4, 7.5.